already watched it, or it will be useful when you watch it. So please ensure that you follow through, and then we'll see. I hope you can see the screen now, right?
Yes, sir.
Yes, good. So what is an episomal vector? That is what I'm going to discuss. As I discussed before, what is an episome? You can have a cell, and the cell is not, just give me a second, please. Give me a sorry. Yes, I was talking about an episome, and first thing we need to define what is an episome. Assume that this is a cell, and here is the genome of it. Assume this is a bacterial genome, or it could be eukaryotic as well. And you may have a plasmid, something like this, existing as a separate entity from that. It is not integrated into the genome. Whereas the same thing can also happen where you have a cell and a genome is there, which is something like this. And into the genome, the plasmid or whatever, plasmid or something has integrated like this. Then those parts are referred to as are integrated. Okay, they are integrative plasmids and phages, as such as prophage and other things. They are integrated. Anything that is existing as an independent replicating entity within a cell or nucleus is referred to as the episome. If something has to exist as an episome, the important things that are required are we require that it should have an origin of replication. So the origin of replication should be compatible with the cell. So if the regulatory or the initiation elements of the cell that the cell has, they should be able to recognize this origin of replication and start initiation. Okay. And as I discussed before, any eukaryotic the vectors of eukaryotes usually have two sets. All of manipulation is done in E. coli, as is shown. Majority of them are done in E. coli, and our application is in other cells. Say I'll write it as eukaryotes. Okay. So we should be able to propagate within the bacterium as well as within the eukaryotes, and it should be a special as a separate entity. So you should have an origin of replication for E. coli, so that this will ensure that there is replication from E. coli within E. coli when this plasmid is or vector is within E. coli. When it is transferred from say this is a bacterium, okay. And now we have in a eukaryotic cell, and we have isolated this, and we are putting into the eukaryote, which is having a nucleus. Assume that this is it, and the episome is existing separately like this. When the vector is existing as a separate entity like this, then it is an episome. So you need origin of replication for that. Okay. And I think here is a selectable marker. BLA is for beta lactamase. Selectable marker is for TRIP. That means the host you know what is auxotropy? Yes, no, you can type in the message and I'll see if you need a discussion on that. I think in the video I have described it well enough. You should still want to say this is a cell and it has all the genome. It is required, all the chromosomes and genes are present. Okay, but it has deficiency in synthesis of tryptophan. That means some genes involved in the synthesis of tryptophan, biosynthetic gene, is deficient, or the gene is mutated. Therefore, this host is TRP minus. And if you take it and try to grow it in a medium containing, that is minimal medium, where you're not giving any complex molecules, but you're only giving all simplest, like such as ammonia, glucose, or something like that. Okay, um, ammonium sulfate, like those molecules, inorganic kind of molecules. Except the organic molecules probably is the sugar that you're giving. Okay, no amino acids, no proteins, nothing of those sorts, no lipids, and so on. So the bacteria, the cell has to take in all those basic compounds and then synthesize organic compounds such as amino acids, which will then be used to make proteins and so on. So this host cannot grow in minimal medium because it is unable to synthesize tryptophan. And tryptophan is an important amino acid for the synthesis of proteins. Whatever proteins this might be synthesizing, they are all non-functional because tryptophan is missing in all of those cases. Or translation might not happen at all. But if you have supplemented minimal medium with tryptophan, then this cell would be able to grow. Why is it able to grow? Because it will take the exogenous tryptophan into the cell and then will use it for its growth. Okay. So what we do, we use this as a selection marker, tryptophan. So what we do is, the cell is TRP minus. We are having a plasmid. On the plasmid, we have the TRP, kind of, you can say, as TRP plus. That means the gene for the missing gene here. The biosynthesis of tryptophan requires multiple genes, not just multiple enzymes. Okay, so even if one enzyme is missing, then synthesis of tryptophan will not occur. So what happens is, in this case, there is one tryptophan. Say I'll call it as TRP1, just to make it along with this. TRP1 is missing, so you have that on the vector. So now we will do transformation or transfection. So we take cells. I will just draw again separately here. Let me erase this off. So for understanding sake, we will do it. We will redo it. These are cells, and they are TRP minus. And you put them in minimal medium, no growth. You take cells in minimal medium, supplemented with tryptophan amino acid that you are already providing readily synthesized tryptophan. Then growth is observable. Now what you are doing is you are adding the plasmid, which is having tryptophan gene, and you are having the cells. That means you're doing transfection. Okay. And when you do transfection, some cells will grow, some cells will take up the DNA, some cells may not take up the DNA, may not receive it. And this whole mixture, you're going to put it on minimal medium. So all those cells that are that did not receive the plasmid here will not grow, right? These will not grow because they are deficient in preparing tryptophan. Whereas those cells that have received the tryptophan, the plasmid, which has the tryptophan gene, they will be able to grow. So that is how you can use it as a selectable marker, because it will eliminate all the non-transfectants. Okay, so that is how you can use an episomal vector. While we keep seeing the next ones we will understand more aspects. I think this is the same, so I'll go here. Here is a different one, just that there is an addition to it. Here is a centromere vector. I mean, in the previous one, what may happen here is when one cell, this cell, for example, if it divides or undergoes binary fission, there is no guarantee that both the cells will receive. One cell may receive and the other cell may not receive it. So how can we ensure that all the daughter cells inherit the plasmid? We should have done something that is by addition of a centromere. Centromere, I hope you remember, during cell division, this is the centromere is the middle part, right? You have a chromosome like this, metaphase one. Here you have centromere. Centromere is the place where kinetochores are organized. That is, kinetochores are some multi-protein complexes like this, which also include, to which also the motor proteins are attached. And these are bound to the spindle fibers like this. Okay, and they are in the one of the poles like this. And during anaphase, the protein between these two, that is cohesin, breaks up, and then these two centromeres are pulled together. I hope you can. The next stage would be something like this way, right? The chromosome, the chromatids, sister chromatids are now split up, and they will be pulled to the opposite poles, which is likely to become a new cell in the future. So the role of centromere is that it is ensuring that each of these chromosomes are inherited to the daughter cells. When the daughter cells form, each of them will get these three chromosomes. I'll just put it like this, okay? It should be 4. You will not have an odd number, isn't it? Unless it is in meiosis. If it is mitosis, it should be even number all the time. Think upon that also. So if we want this faithful inheritance of a genetic material to the daughter cells, we should have something like a centromere. So in this one, the rest of the things being same, they have added a centromere, and ARS is an autonomously replicating sequence. Okay, it's just another name for kind of origin of replication. I hope this is clear enough, right? So because centromere is there, even if it is something like this, the spindle fiber might form like this, and then they will be inherited to the to each of the poles. Okay. Then there is integration. Here until now we have spoken about something like which will exist as an episome. That means it does not integrate. Now how can we have an integrative plasmid or integrative vector? Here is one example for plasmid. The selectable marker here is uracil, a gene encoding for one of the biosynthetic genes for uracil. So the cell, the host that you have should have chosen should be, the host should be uracil 3 minus. That means, again, as in the case of previous one, I will try to describe so that you'll understand. If you take a uracil host like this and you grow it in minimal medium, it will not grow. But if you take host uracil 3 minus in minimal medium plus uracil, then it will grow. Okay. So that is why what we have done now is into the vector. I first want you to understand that there is only one origin of replication. There is no second origin of replication. It is because we don't want this plasmid to exist as an episome. we are kind of enforcing that any cell that is growing should have integrated this plasmid. Okay. So now if I put if I'm having uracil 3, the functional gene, on the plasmid, and on the host we are choosing something that is uracil 3 minus, okay, gene for one of the genes. So now once you think of it like this, we will take different scenarios for your understanding. I hope it will enhance the understanding. Let's see. So now we have 3 different situations. We have added this plasmid. Okay, assume this is the plasmid. We have tried to do transformation. In one case, it is integrated. It is like this. In this case, it is not transfected. It did not happen. Okay. And in another case, what has happened is that it has got integrated. So this is the plasmid integrated. In another case, like this and like this. What will happen when we put this in minimal medium? That is how it's a way to think of. If we have it on minimal medium, which of these will form a colony and which one will not form a colony? It's always important to think in this way so that you will not miss out on possibilities, and you will be able to zero in on the highly probable situation that might be happening. So in minimal medium, we will give names 1, 2, and 3 situations. Situation 2, where no DNA was inherited, no vector is inherited, it will fail to grow because it is uracil minus. The host is uracil 3 minus, therefore it cannot synthesize uracil, because of which it will die. These 2 cells will grow. Which ones? Situation 1 and 2 will have growth because they have inherited the DNA. But which one will form a colony? You can think of it like that. Which one will reproduce and form a colony? What is a colony? A colony is kind of almost identical cells which have the same genotype. You can also think, we will have a little bit discussion on this one. Okay, assume that this is the cell, and this cell has divided into 2 cells. Now, which cell will get this? Will both the cells get the plasmid I'm talking about? Will both the daughter cells get this, or only one? So let me draw this here. It is existing as an episome. Will both the daughter cells get, or only one daughter cell will get? Only one daughter cell will get, because because there is no origin of replication for this plasmid. There is no origin of replication to to be replicated within when it is in yeast. You have one that is only functional when it is in bacteria. It is not functional when it is in yeast, right? So only one of it will get, and that the newly produced cell did not inherit any of this vector, because it has not replicated. Then what would happen to the newly formed cell? It would die out, because it cannot. It's now uracil minus. It cannot by itself synthesize whole of uracil. Okay, so if you think of which ones will form colony and which ones will survive in minimal medium, this will survive, and also this one. Which will form a colony? This will not form a colony, because it will only remain as one single cell. Again, if this divides, one will inherit the vector, the other one will die, right? It's kind of it's not going to happen. So now we have the third scenario, that if this cell has divided, then you have the cell, and within the cell we have this vector, sorry, nuclear DNA or the chromosome, and it has the vector. Now, if the cell um, integrated plasmid. Now, if the cell divides, so it will form two colonies, both of which, one second. So the daughter cells will have, both of them will have inherited the plasmid, right? I hope you can see that. Why is it inherited? Because it's already, there is now a possibility of replication because it is already integrated into the chromosome. Although I drew it circular, it is not, but still you can think of it. All the chromosomes have origin of replication, and the origin of replication, because of which the integrated plasmid also replicates, and both the cells, daughter cells that are produced, are uracil 3+. So they can produce uracil. Okay, and they will form a colony. That is how you have to see this thing. There is one, how integration happens is a question. The host cell that is present is having a mutation of uracil in that one gene. You have used an identical or exactly almost the same copy, uracil 3A, only that this is functional and this one may be having one or 1 point mutation or something like that. Now, because of these two sequences are homologous, in the sense they have large areas of similarities, this one and this one. Therefore, a holiday junction will form, and it will be resolved, ultimately resulting in the two copies. This part is having the non-functional uracil gene, and here you have the functional uracil gene, and everything in between, all these things will get integrated here like this. Okay, that is we are, by not adding an origin of replication for the yeast, we are now, when we select, we only select those that have successfully underwent homologous recombination. Okay. Yes. If you have any questions, you can post in between so that we will know. I'll be able to speak those details if required. Second one is the Costsite. It's all same except you're adding a Costsite. Why do you need to add a Costsite? What is the need for Costsite? The size of a plasmid is limited, in the sense when you do transformation based on the size of the plasmid, size is, I don't know if I wrote it, inversely proportional to the efficiency of transformation. Okay, we are usually using chemical or normal, if transformation kind of thing. The larger the size, it will get difficult to put it into the bacterial cell, because the time taken for it to enter the cell will also increase, and because anyway the size is there, a lot of issues may happen. So to overcome this, our problem is not about the plasmid. If it has already, even if it is large and it has entered into the cell, it can survive for it can replicate, because it will have an origin of replication. It is circular, so it will be kind of stable enough at least. But then the problem is how, if we can manage to put the plasmid into the cell, how can we make this entry happen? We know from what we have learned about the basics of phage biology, they have a kind of injection system where they infect, they make a hole, and they inject their DNA inside. Then we should be able to, we don't need all the phage genes, because they occupy about something near around 30 kb is occupied by themselves. When you see a majority of the lambda vectors, you'll see up to 30 kb of fragment results is required for genes of structural genes, genes involved in replication and other things. Now here comes a plasmid, which is a bit between cos, that is Costsite, cohesive site, and the plasmid, right? You take this and here, so you add a plasmid. What we need for its existence is just origin of replication, one selectable marker. That means if you have 50 kb of lambda vector, if it is a phage one, lambda vector, then 30 kb is occupied by it. The maximum size you can clone is only 20 kb of the fragment. What if we see the plasmid, it can be very small, something like 5 kb. That means 50 minus 5, assume you can clone up to 45 kb in it, right? So if we want, the best way is to somehow be able to package the head particles, package this plasmid into the head particle. As we learned in the in vitro packaging, or how packaging happens in phages, there is a protein which recognizes the Costsite, and once it recognizes, and then it say, I hope you remember the concatamers, what I meant by concatamers, right? This is assume Costsite, another Costsite, another Costsite, like this. That is a concatamer. Each of it is a lambda genome. When we discussed about it in the introduction to vectors, I think I described those things. So what it does is, the protein that is present here, packaging protein, it binds, it recognizes the Costsite that is here. And once it recognizes, and then it will push the rest of the DNA into the head particles like this, until a second Costsite has encountered. So assume that this is the concatamer, something like this, with lot of Costsites. So this is the first Costsite, here is the second Costsite. Once the packaging protein comes in contact with the Costsite, it will push the rest of the DNA into the head particles until it encounters a second Costsite. Once the second Costsite is reached, what it does is it cuts off here, and then this head particle is packaged as one phage particles. It will give, and the neck, stalk, and other regions are tail fibers are all made. Okay. So if we want, now the idea is that we want it to package. So for packaging to happen, we need a Costsite, and therefore in this plasmid they have added a Costsite. Now what happens is, you can clone a very large fragment in BAMH1, even if it is large. It will be, as I said, I'm not going into all the details, but there will be a lot of things. This is required there are a lot of other processing in between, and to have something like a concatamer, or even if not, at least presence of one Costsite is important for the packaging, and then it will be packaged. So that is the reason why we are adding a Costsite. Okay, to increase the the objective is to have a larger fragment. If you want to, if you increase the fragment size, there is a difficulty in introducing the plasmid vector into the bacterial cell. So if you want to introduce, we can use a system like that of phage, which can inject the DNA. That is much more efficient. But if you want to put the plasmid into the head particles, you need something like a Costsite. Okay. Good. I hope you understood. Then the next one is for, did we finish this? I'm not going to discuss this much, but it is simple in the sense you have, it's for gene expression in mammalian cells. So you should have chosen a promoter, something like promoter of cytomegalovirus and a promoter of simian virus, the origin of replication of simian virus, so that it will, simian virus can exist as an episome within the cell, right? This is, assume this is the nucleus and you have a lot of chromosomes, and then the simian virus can exist as an independent entity like this. So it's able to, because, so what people have done is put the origin of replication into this SV14. Then we need a gene expression to happen. So there should be a promoter that is very active or strong promoter. They have chosen one from cytomegalovirus, and here you have the multiple cloning site. You would have cloned the gene of interest here, and then it would have produced. There is, as I was describing in the video, there are certain things for the eukaryotic mRNA, especially in the mammals. It has a poly-A tail, it has a G-cap. Majority of the things that are transcribed, G-cap is added by the enzymes. And then you have a poly-A tail that is also added to the mRNAs because there is a poly-A signal. If we want something to be expressed in E. coli, the G-cap and poly-A tail are important for stability and also translation, right? So that the protein forms. So we need to, G-cap will be added by the cell. We don't need to do something separately for it, but we need to put a poly-A signal, polyadenylation signal, like as in here, so that the mRNA that it produces of the cloned gene would be something like this. And then translation would occur and a protein of interest is produced. Okay, I hope I covered enough things in this. Then about lambda phage, should we discuss this? I think I already discussed this thrice. I want to see if there are any others. I would want to check that. Yes, I have, okay, 18 minutes. I will go with this one. And please watch the lambda EMBL3A once more. I have already discussed this twice, so I'll skip this at this point. If required, I will take it up in the next class. So there is something called as M13 phage. It produces single-strand DNA. It is a single-strand DNA when it is in phage. Upon entry into the cell, it becomes a replicative form. There are two replicative forms. It becomes double-stranded. It is actually a single plus strand, and then the second strand, minus strand, is also synthesized. So it can exist as a something almost similar to that of a plasmid. Okay, and from this, new plus strands can be synthesized, and it forms a head particle enveloped virus, and then it buds off the cell. It doesn't kill the cell. So there are a lot of advantages or in some cases we need to have these kinds of vectors. First thing, single strands are useful for multiple purposes. One is to generate probes. There is second one that is for site-directed mutagenesis or random mutagenesis. You can say, I'll just put it as mutagenesis. And there is one more, what is that called? That's for sequencing. Now, nowadays, we don't need all this stuff. We can do it in much more sophisticated ways, but it's important to know all the genetic elements or genetic gene elements that are available to us so that we can manipulate in the future cases. So for this to occur, it's simple, right? You can, why M13, why I want to discuss about this is simple. Say this is happening within this cell, okay? This part, it is single strand like this, and then you have in the cell it forms a double stranded. Very similar to what we would have done for plasmid isolation. You can, it almost behaves like a very similar to a plasmid. So we can isolate that, and we can just do the cloning or making a ligation, making a recombinant, and then put it back into the cell, similar to that of transformation. And now it will generate a plus strand, and you get the phage particles. All the phage particles are single stranded. Now you just take them and do your analysis. What you want to use it as a probe, or you want to use it as use it for mutagenesis or sequence. You can directly use it. You don't need to separately synthesize a single strand. The phage is doing for you, and it is. I think it seems much more convenient in many aspects for this reason. The ease with which we can isolate, do the manipulations, and put it back, and we directly get single strands, right? So that is a nice way of. That's why it's a nice vector. So I'm going to give you a question. I actually had a few questions, but I thought of taking this one, artificial chromosome. So I want you to do this one as well. Lambda zap. These are the things that you have to do self-study. You have there are different types of vectors, and their capacity is we keep on increasing. One of the objective is to generate vectors that are high capacity vectors. That means we should be able to insert a large fragment. Especially at one point, we wanted to sequence genomes. Genomes are huge, and if you want to do that, if you are going at level of 25 kb or something like this, it will take ages for us. So people wanted to do clone larger fragments, and that is when these bacterial artificial chromosomes and yeast artificial chromosomes also have come up. So your self-study part is here, that you should know the advantages and disadvantages of each of these. Okay. Even yaks have some disadvantage, backs and yaks. So you should try to know what are the advantages and disadvantages. And there is one important vector. I would want you to know what is lambda zap. Try to know its structure, the components and features, and its advantages. It's a very interesting vector. Please try to find out what it is. And there should be one more that I wanted to discuss, but I'll not waste time. I have a question that is taken from the previous question paper. So please try to solve this fast, and I need you to solve this fast and upload it. Upload the answer on the stream itself. Okay. You have 12 minutes to finish this. Please do it fast. You can also turn on your mic and tell us what's the mistake, or what are the problems associated.
Sir, I have a doubt. Can I ask now? Ask
Yes.
later on.
Yes.
Okay. So
Ask.
when you use alkaline phosphatase and it converts the phosphate to a OH, how do you convert it back to a phosphate so that ligation can occur?
In which case are you speaking?
So no, before when you said that the when adapters are attached, before attaching, alkaline phosphatase will be used to prevent the formation of dimers. So in that case, when
Yes.
it has to get ligated to the vector, won't it affect it at that point also because it won't be readily attached?
Okay. I understand your question. So this is the fragment. Okay. And assume this is the adapter. Meanwhile, others, I please work on this problem. Okay. It is related anyway. So assume this is the 5 prime, and this will be the 5 prime and 3 prime. So you have OH here, and this is OH, and here you have OH, and here you have a phosphate group. Here you would have a OH, and here you would have a phosphate. So if do we need to, what we are doing right now, when we add ligase, it will seal these two like this, right? And now we want to have, we will isolate this fragment. Okay. Let me erase the rest. We will take the same. Now we have phosphate. These two are sealed like I've shown in this case. Now we have OH, and here also OH. We are now treating using PNK, polynucleotide kinase, which will put a phosphate group here and a phosphate group here. Now we will have the vector. Vector would have the complementary overhangs. So I'll put it like this. Right? What do the vectors have? The question is, this is 3' OH, and here is the 5' phosphate. This would be the 5' phosphate here, right? And this is the, I will erase the rest. Why should we need to use PNK and phosphorylate it? That is one of the questions. You would have, one second.
Yes, I kind of got it now. It's to
Got
prevent
it?
having two OHs on both of the sides.
Yes. Especially this one is important for one reason. If the vector is also treated with alkaline phosphate, I think I'm telling some answer, so I shouldn't want to describe more, I guess.
Yes, okay.
Yeah, if I've treated, okay, let me answer it anyway. If I treated the vector also with alkaline phosphatase, this will not be there, and here this phosphate will not be there for the vector. And if I haven't treated the adapters with PNK, okay, assume I did not treat the vector adapter used, where we have added the adapter, we did not use the PNK. Okay, we will take the adapter as it was. What was it on its 5' side? OH.
OH.
On 3' side, OH. Here, OH. And we treated the vector with alkaline phosphatase, and also the adapters do not have 5' OH. Here is OH, here is OH. And this is OH. This one is OH. If we add ligase, what can it do?
It
Ligase
won't get ligated.
ligase cannot do anything at all to this kind of situation.
Yes.
Okay, therefore we should have, if we are either, the rest of the things I think you can figure out from the question is highly related. Try to answer this question. We have 5 minutes. Critique the experimental setup. There are, I think I made this with 2 or 3 mistakes in it. I hope you can try to find it out. Technical mistakes, yes.
Okay, sir. Thank you.
Yes. Um, I would also want to draw your attention to this one thing before I think we are close to closing the lecture. So, if we are treating with alkaline phosphatase, this is something you need to remember. If we added ligase to this, what would happen? Ligase will be able to seal. Sorry, ligase will be able to seal here because there is OH, 3' OH, and 5' phosphate. So it will seal this gap, right? And but it will not be able to act on this side, right? But if this was a complete molecule and you put it into the cells, if you transformed it into E. coli cells, the repair mechanisms will take care of it and will seal this gap. That means they will do the repair part, they will remove some bases and fill it up and make it continuous. So it's not always essential that we have to treat it with P and K. But P and K, it all depends on where and when you're treating something with alkaline phosphatase. As in this case, if you remember, usually the vector is treated with alkaline phosphatase because we want to reduce the number of religation. And we are already having in this situation where the adapters are having are 5' are treated with alkaline phosphatase to remove the phosphate group on the 5' side. So if you want to have ligation to, if you want ligation to occur, we should have put that with phosphate. We should have restored its phosphate group so that the ligation would occur here and here. You can imagine that I hope you can see that these two, it's not sealed here and here. But even then, if you transform it, it can be repaired within the cell. Okay? Try to remember that. And in this question, the biggest flaw is that she has treated it, she or he, it can be, treated with, where is it? So she treated the vector and fragment with alkaline phosphatase. Then she would perform ligation reaction. Ligation would not have occurred at all. And then the predict the result. Yeah, you would not see any colonies grow technically. And the mistake is that both are being treated with alkaline phosphatase. The best thing is to choose either vector to treat it with alkaline phosphatase or the fragment. Typically, it's always the vector because we want to avoid religation. Okay. And is there any other mistake in here? Okay, that should be all right, I think. Yeah, these two are isochordomers, BAMH1 and SAU3A. Is there any? Can you identify any mistake? Any other mistake? We have. Yes. Yes. Here, this vector is digested and fragment is there and then treated with alkaline phosphatase. So the best question is, one of the answers is to put back the phosphate group using P and K. The best, if you're critiquing the experimental setup, you should say you should not have used alkaline phosphatase on both. It should have been mostly the vector, and fragment should not have been treated with alkaline phosphatase. Okay, in the next class, we will discuss further details. Please, I hope you're all following, and we should be in touch. Ask questions if you're falling out, and if there is something to be repeated, please bring to my notice at the earliest. Don't wait until some other time. Okay, this is the end then. Take care and have fun. watch the videos as soon as possible, please. Okay, thank you.